The trial I think you're referring to is the one looking at azacitidine plus Romidepsin. And that's a clinical trial that goes back to some early preclinical work we've done looking at trying to identify interesting doublets and hopefully soon triplets of drugs that might have interesting activity in patients with T-cell lymphoma. As most people are aware, CHOP-based chemotherapy, most experts in the field feel is suboptimal treatment for these patients, and we really need to find newer strategies to try to overcome the limitations of CHOP-based chemotherapy. We can do that by adding drugs to CHOP, or we can do that by identifying novel platforms. And this particular effort that you're referring to is an effort to define a new platform. Now, interestingly, the T-cell lymphomas seem to be diseases characterized by lesions in various epigenetic operations. And we know that there are a number of epigenetic defects in peripheral T-cell lymphoma and angioimmunoblastic T-cell lymphoma that carry mutations in DNMT3, IDH2, and TET2. Sort of the collective effect of those mutations is that they alter the methylation status of a whole variety of genes that may help contribute to the misbehavior of T-cell lymphoma. Romidepsin, of course, is an HDAC inhibitor approved in the disease already. So working under the hypothesis that maybe these diseases may have a particular vulnerability to epigenetic drugs, we explored in various preclinical models, a paper recently published by Enrico Marchi from my group, that the combinations of hypomethylating agents, decitabine and azacitidine, were really remarkably synergistic with HDAC inhibitors. And it really didn't seem to matter what HDAC inhibitor or what hypomethylating agent, but these seem to be class effects in terms of the synergy seen. So we translated that to a recent phase 1 study, and that's a poster we're presenting today, in fact. And clinically, we've now treated 20-something patients. The majority of them appear to have B-cell lymphoma, but 5 of those patients have had T-cell lymphoma. Of the 5 that have had T-cell lymphoma, 2 have seen prior Romidepsin. And all 5 of those patients with T-cell lymphoma have very heavily treated disease. So they failed all of the traditional chemotherapy options. And of those 5, 4 of the patients with T-cell lymphoma to date have responded, and 2 of those have been PET-negative complete responses. So it's very early. It's hard to make grandiose conclusions about the activity of that particular doublet in patients with T-cell lymphoma based upon only the 5 patients. But what's interesting is we really haven't seen much activity in patients with B-cell lymphoma. So even though the majority of patients have B-cell lymphoma, we haven't seen activity. A lot of the activity seems to be concentrated in patients with T-cell lymphoma. It's 4 out of 5, but that's 80% of the patients, in contrast to maybe less than 20% of the patients with B-cell lymphoma. So we think there may be some lineage specificity of this combination that allows us to try to use it to take care of patients that might have chemotherapy-resistant T-cell lymphoma.